welcome to another interesting conversation on a raging topic that is taking over the globe. The scientific community in particular is debating this very actively, and that is the origins of the coronavirus and all the theories that have come out, including the leaks, the email leaks of Dr. Fauci, who is considered to be one of the foremost spokespersons in the world when it comes to this virus. I'm delighted today to be joined by an expert of our own, Dr. Abdul Ghafur. Welcome, Dr. Ghafur, to the show, and really I want to thank you for sparing the time. Just a little introduction about Dr. Ghafur. He is a consultant and adjunct professor in infectious diseases based out of Chennai in India. Dr. Ghafur is a renowned expert in the academic, policy, research, and political aspects of infectious diseases. And we want to know a lot about the political aspects of infectious diseases, as a lot of politicians around the world are commenting now. He's a clinician with an MD and MRCP, and also a researcher with an additional qualification of FRC Path in chemical microbiology and virology. He has had the honor to be interviewed by the British Medical Journal for the BMJ Confidential as recognition of the work in the antibiotic policy field. He has delivered lectures on numerous prestigious international infectious diseases and health policy conferences around the world, and has been published in well-reputed journals such as the British Medical Journal, Lancet, amongst others. He is an editorial board member and reviewer of many international journals. He received the Indian Medical Association Oration Award for his contribution towards tackling antibiotics resistance efforts. He is a member of several health policy committees at the national and the international level. Thank you so much, Dr. Abdul Ghafur, for joining us this afternoon, this sunny Monday afternoon, in what happens to be the second year of this pandemic. And thank you once again for joining us.
Thank you. Good afternoon.
Good afternoon to you as well. And we might show this in the evening or the night, depending on how long our editing takes. So greeting of the day, whichever time of the day people tune in to watch this. Dr. Ghafur, just to get like the song says, says that let's start from the very beginning, because the beginning is a really good place to start. So when it comes to Corona, my own layperson perspective, because I like to track these things, was that in December 2019, I heard about this unique fever of sorts that was taking over this particular province called Wuhan in China. And I recall speaking to a senior journalist friend of mine, who works a lot on the defense beat as well, and asking him about this virus and saying, what are the prospects of it getting to India? And, is it some sort of SARS? Because 18 years ago, the SARS that took place was really as pandemic, as close to a pandemic we got in living memory. And he said, no, this has to do with, it's a localized thing. It's some wet market. It has to do with something that's being eaten. It's going to not survive in a hot country like ours. And I kind of was satisfied with that answer till, of course, everything exploded around us. So, Doctor, let's start from the beginning. When did you hear about this particular virus? I know in your work, you must be tracking this day by day.
I heard about this virus probably in the first or second week of January, when we heard about a new coronavirus that has originated in the Wuhan city of China and that's spreading in China. Initially, we thought there is no human-to-human transmission. That means if one person gets, other person will not get from the first person. And we were all so happy and relieved. Oh, it's a new coronavirus. But don't worry, there is no human-to-human transmission. Over the course of the next 2-3 weeks, everything changed. The Wuhan was in trouble. The Wuhan was shut down, and people were returning from Wuhan to other countries, including India. And then the explosive pandemic started. Pandemic impact. Millions and millions of poor people are pushed into absolute poverty by the pandemic. The financial impact across the world has been devastating, much more than the Second World War. Isn't that a horrific period that we are experiencing? Our personal life has changed, social life has changed, The global life has changed, the world has changed, and we need to discuss what exactly happened.
Yes, absolutely. I'm a little surprised that there aren't more discussions on what exactly happened. It's picking up now, but I remember reading that I think there was a Senator Tom Cook from Arkansas in the United States. Who was one politician who was talking about this back in January 2020, and he was kind of pushed into silence. He wasn't covered by the mainstream. Of course, it was an election year in the United States that year. In around the middle of January 2020, the WHO also said that there was no proof of human to human transmission, which again, I think kind of made the rest of us feel far more comfortable with this. Before, in many ways, it was too late.
You know, in the beginning of the pandemic, we heard stories of lab origin, and we all believed it's all conspiracy theory.
Right.
The scientific world believed the virus originated from the wet market in the Wuhan city. And we believed that. We had no reason to disbelieve it. Then over the time, we started reading publications in the scientific journals and preprints, in the academic discussions on a possibility of laboratory origin. But they were all just possibilities, because the world famous virologists that people like me respect refuted those theories, and we believe them, and we still believe them because they are well respected people. But still, over the last few months, there have been additional publications and discussions, even in reputed journals like the Science magazine. Recently, 18 scientists, highly respected scientists, wrote a letter in the Science magazine to open a discussion on the both possibilities of the laboratory origin. And they are all respected scientists. We have no reason to disbelieve them. Many of them we know very well, and they are respected. They are men of science. They are not conspiracy theories. When they openly say, we need an open discussion on the possibility, we are not, nobody is telling it is laboratory origin, but we want to discuss a possibility of a laboratory origin, and we need to discuss that. Even World Health Organization had an inquiry into the Wuhan for the possibility of natural and laboratory origin, but the conclusion was a laboratory origin is very, very, very unlikely, and it's most likely a natural origin. But now there is more and more emerging scientific evidence that laboratory origin is a possibility. It may be a remote possibility. I'm not saying it is laboratory origin, but now scientific world has started the discussion that there's at least a possibility of laboratory origin. And why do I come forward to discuss that? It's a controversial topic. It's a contentious topic. Why do a person like me, who is a clinician, a researcher, and a policymaker coming forward to discuss the issue? Because, and I'm coming forward on my personal capacity, and has no direct or indirect relationship with organizations or institutions or the committees that I am associated with. Why do I come forward? Because even if there is a remote possibility of a laboratory origin, we need to discuss that remote possibility. Because if it is true, such incidents may be repeated in future. We need to prevent the remote possibilities of such future incidents. Scientific community do not have to prove or disprove laboratory or natural origin. What we just need is, we need to come forward in a very open, non-critical, unbiased way, in a very diplomatic way, without any finger pointing, and discuss the possibility of a laboratory origin, and initiate measures at international level, at United Nations level, to find a solution so that such remote possibilities are not repeated for our future generations. Let me quote an example. The nuclear weapon. We do know it exists, but United Nations came forward and created a nuclear non-proliferation treaty signed by all countries. And how did that happen? Because the scientific community who created the nuclear weapons and nuclear technology came forward and convinced their leaderships, convinced the United Nations on the need for such a nuclear non-proliferation treaty. Are we blaming the scientists who created the nuclear weapons? No. Otto Hahn is a German scientist who invented, who discovered the fission technology. It was Robert Oppenheimer, the very famous physicist, who led the Manhattan projects, which ended in the creation of nuclear bomb. It was Albert Einstein, the godfather of science, who wrote a letter to the American president at that time, asking him to initiate the Manhattan Project for creation of nuclear weapon. Are we blaming Albert Einstein? Are we blaming Oppenheimer? No, they are all still respected scientists. But later, when they understood the damage done by nuclear weapons, they came forward, including Einstein came forward and criticized the nuclear technology, the wrong usage of nuclear technology. And that resulted in the nuclear, they later resulted in the nuclear non-proliferation treaty. We did that for survival of humanity, for the benefit of our future generations. The same thing may be true on the virus research field. Let me give you some background research. The virology community, they are expert scientists. They manipulate viruses, they create new viruses for the benefit of the humanity. I will compare with the nuclear technology. Don't we think we use nuclear technology for power stations?
Yeah.
Just like that, the virologists, my fellow virologists in the scientific community, they combine, they do research on the virus. They cut and paste viruses, create chimeric viruses. For what? For research. And why do we need that research? Because such combination, such cutting and pasting of the genomic material, genetic material happens in nature and then spill over. That's how Ebola originated. The Middle East coronavirus originated, SARS-1 originated. How can we prevent such pandemics or epidemics? We have to get ahead of the nature, create vaccines and drugs for future spillover. That is the ethical aspect of the gain of function research. What is gain of function research? Where scientists manipulate or modify the genetic component of the virus, make it more infectious or more lethal. Why do scientists create such Frankensteins? To study, to find solutions like vaccines and drugs. That is the ethical aspect of the gain of function research. But nuclear technology is like that. Nuclear technology has got good role. At the same time, nuclear weapon can wipe out the world. Just like that, if not properly used, gain of function research can wipe out the world.
Doctor, what is the take of the international virologists community as a whole when it comes to investigating this further for the origins of the virus? Is there a unanimity or is there a dispute at the present time?
Until recently, the international virology community and the scientific community was largely silent on the origin of the coronavirus. Especially, they were hesitant to discuss even the possibility of a laboratory origin. That is not unexpected, because if it is a laboratory origin, who created the laboratory virus? The scientists, the scientific community. If you accept that, it's not just one scientist or one laboratory or one country that's taking the responsibility. It's the world of science taking the responsibility. Just like the world of science had to take the responsibility for the nuclear weapon, here we will be forced to take the responsibility. That is not a small responsibility. If you take the responsibility, we need to come forward and find a political solution at the global level. And that is not a simple thing. But scientists are sincere individuals. Scientists work for the humanity. And if scientists do mistakes, mistakes do happen. Because scientists, the men and women of science, are not goddesses or gods. We are human beings. Science is always perfect. Scientists are never perfect. Scientists make mistakes intentionally or unintentionally, mostly unintentional mistakes. That mistakes can have deleterious effects across the world. But Scientists never keep quiet forever. Scientists have a conscience. Invariably, inevitably, they come forward and discuss the truth because scientists want a solution. Scientific community has no interest in blaming the fellow scientist. What the scientific community needs is a solution to a potential problem. Let me talk about the gain of function research again. We talked about the good aspect, the ethical aspect of the getting ahead of the nature and developing vaccines and drugs. There is another aspect. We all know that there are lots of countries with military laboratories with bioweapon research going on. It's an open secret. There are plenty of countries. In my opinion, any country with an advanced military, advanced defense, and advanced science should have a biological weapon laboratory. I'll be surprised if a country doesn't have. Now, who does this research? The scientists. Why do they do the research? Because they are patriotic to their country. Just like nuclear scientists develop nuclear weapons, just like aircraft engineers develop military aircraft, machine guns, tanks, these scientists do their work. They are not bad scientists. They work for their country. Either they work for the betterment of humanity or they work for the military aspects of their country. Their intentions are always good. Either way, you can question the intentions. Why do people work for the nuclear weapon? Isn't it dangerous? But they are working for their country. And we have scientists, all major countries working on nuclear science. Are the bad people? No. They are good people. They are sincere people. But we need to agree and accept that experiments can go wrong. It has gone wrong in the past in all fields of science. It's a responsibility of the scientific community to come forward and at least discuss the possibility of an error that has happened in the hands of the fellow scientists. So we don't know how to incriminate them. We don't have to pinpoint them. We just discuss the possibility and find solutions so that such incidents are not repeated in the future.
Doctor, at the heart of this is China. And China has had a very opaque response to all these queries. In fact, a lot of journalists have tried to enter and have tried, there are some remarkable stories of journalistic perseverance, wherein I think it was a Wall Street Journal journalist who actually made it to the beginning of one particular tunnel, which is supposedly where this virus allegedly I mean, I'm going to use a lot of quotes because so much of it is, speculation right now. But they say that it was in this particular cave where about a few years ago, maybe 2 or 3 years ago, I'm thinking it was 2016 or maybe a little before that, where 3 miners died. Reporting similar symptoms as that of the coronavirus. And there was journalists who actually made it all the way to that cave and then were turned away because it's heavily guarded. So there is that limitation when it comes to dealing with a country like China, which is extremely opaque when it comes to sharing this kind of information, unlike, say, if this had emerged in any other democracy in the world.
Suppose the virus is of lab origin and that has come out of the Wuhan Institute of Virology. Are we expecting that country to accept it because that virus has caused a global pandemic? I don't think, of course, China will not do it. I don't think other countries will also do it. If a mistake happened in the hands of your scientists, whatever way, do you think that country will accept the responsibility? They will naturally try to hide the source of the outbreak. So what happened in China, if it is a laboratory accident or laboratory leak, the response is expected. They have every reason to hide the investigation, to hide the truth. That is a natural response from every country, and China is doing it. I'm not saying it's a laboratory origin. I'm only discussing the possibility of a laboratory origin. And why do I do that? Yes, the scientific world is discussing about, the newspapers are discussing about the corroborative evidence. One corroborative evidence is that at least 3 to 4 scientists were sick in November due to coronavirus-like symptoms. But we don't have a proof that it's coronavirus. Maybe, may not be. I'm not saying no, maybe, may not be. There are a lot of corroborative evidences. the US National Institute of Health, Anthony Fauci and team were funding the Wuhan Institute laboratory for coronavirus research. He has never accepted. He funded gain of function research, and Peter Daszak was mediating it. But let me put another perspective. It's only for a perspective. I'm not telling who is right or wrong. Collaborative work is the basis of scientific progress. No science, no scientist, no laboratory, no country develop any scientific research, scientific development by its own. All these works are collaborative research. So we don't know the level of collaborative work they did. We believe everything was ethical and for the betterment of humanity or for the betterment of their military laboratories. We are not questioning that. But whatever happened is a collaborative work in whatever level. But that can be justified, at least from their perspective. These are all collaborative evidence. Now, where will you look for the real evidence? Where will you look for the real evidence whether a virus is natural or origin? You have to analyze the genetic component of the virus. That's what we need to discuss here, because the public should understand the basis of a possible, it's not an absolute proof, a possibility of a laboratory origin. So let me explain that. The virus, we believe, either has come from the nature. And how does a virus come from nature? As I explained before, the nature is full of various kinds of virus. And we all know that the genetic components of virus mix up in animals. And new viruses are created, always. Because nature is the ultimate vessel. Mother Nature is the ultimate creator of the life, and Mother Nature is capable of doing everything. So what we believe is that this SARS-CoV-2 virus that caused the COVID-19 originated in an animal probably called as pangolin. What happened is the pangolin has a coronavirus that got mixed up with a bat coronavirus. There's a special type of bat called as horseshoe. If you look at the face of that bat, it looks like a horseshoe. It's called horseshoe bat. There's a horseshoe bat virus called as RATG13. 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 That entered the pangolin's body and mixed up with the pangolin's coronavirus and created the SARS-CoV-2. And from the animal wet market in Wuhan, it jumped into the humans, and from one human to other humans, one place to other place, one country to other country. That is the typical natural origin theory. Now there is a laboratory origin theory. Some people believe this virus has characteristic of a laboratory engineered virus. And why do people believe that? I will go into a bit of a technicality of virology and genomics. I know it's highly technical. I will try my best to simplify it. The genetic code is the code of life is written in the genome. For example, human cell has got DNA. That is a code of life. Whatever happens in our body, all the components, everything is coded in the DNA. That DNA has 4 letters. The language of life is 4 letters. A, T, C, G. A, T, C, G. 4 letters. Now, if you make words out of 4 letters and each word having 3 letters, we call triplet, 3 words, triplet codons, codons, triplet codons from 4 letters. How many words you can make? 64. So 3-letter combination of 4 letters, there are 64 words. The code of life has 4 letters and 64 words. And what are these 64? What do these 64 words do? They code for protein. Protein is the material aspect. And protein is made of amino acids. Amino acids are the building blocks of life. So the codes, the 64 triplet codons code for 20 amino acids. 64 triplet codons, so 64 words and 20 building blocks. So you can understand that more than one word can code for one amino acid. Please note down this point. More than one triplet codons can code one amino acid because there are 64 codons and 20 amino acids. You will later find out why I am stressing this particular aspect. So basically, first there is DNA. The DNA gets converted into RNA. It's a different type of genetic material. The letters are a bit different. Instead of DNA, ATCG, here it is AUCG. The same 4 letters in a bit of a difference. So RNA has 4 letters AUCG, and that become amino acids, that became protein. Now there are 2 types of virus, the DNA virus, RNA virus. Our famous coronavirus is an RNA virus with 4-letter genetic code AUCG and 64 word language made up of these 3 letters, again 20 amino acids. Now let us look at the structure of the coronavirus. It is technical, but just imagine a rabbit's head. The coronavirus ball is like a rabbit's head. A rabbit's head, let's do 2 spikes. In fact, coronavirus has too many spikes attached to it. Use the picture, a ball with so many spikes. So let's forget about all those spikes. Let's think about one spike. The rabbit's head is the virus body, and one spike is like a rabbit's 2 ears. Like a rabbit 2 ears. And we call it as a spike. The spike has 2 components, 2 subdivision, like rabbit's ears. The S1 component, because spike 1, and the S2, spike 2. So 2 components, and in between, just like the gap between rabbit's ears, there's a clump, there's a cleavage. We call the cleavage between the S1 component and the S2 component. So we got a rabbit's head, rabbit's ears with a cleavage in between. Let's look at one of the rabbit's ears, one of the S1 component. The S1 component has got a very important part called as receptor binding domain, RBD. Why is this RBD very important? That is the one which first gets attached to the human cells. And human cell has got a receptor called as ACE2, ACE2, angiotensin converting enzyme 2 type of receptor. That is a receptor on the human cell. For example, human respiratory tract has got so many cells with ACE2 receptors. If the virus enters the human nose and respiratory tract, the spike protein, the S1 component, the receptor binding domain of the S1 component gets attached to the ACE2 receptor of the human cell. Once that attachment takes place, the S1 and S2 gets attached to the human cell wall, cell membrane, and then enter the human cell, and then they start using our machine, our factory, and start making babies. So that's why the receptor binding domain of the S1 component of the spike protein of the virus is very important in our discussion. Now, the natural origin theory believes, apart from the receptor binding domain, now the overall the coronavirus SARS-CoV-2 has got 30,000 letters. I told you about ATCG or AUCG for a RNA virus. There are 30,000 letters in the genome, in the RNA of the SARS-CoV-2 virus. Of that, receptor binding domain is around 400 or 500, around 400 letters. Rest everything, we can call it as the backbone. The different structures, I'm not entering the detail. Let's call it as rest everything as the backbone. Natural origin theory believes the backbone is made of RAG13 bat coronavirus. And the receptor binding domain is made of pangolin's virus receptor binding domain. So what happened was the bat, horseshoe bat RAG13 virus jumped into the pangolin's body, cut and pasted the receptor binding domain, because cut and pasting happen in nature. Nature is very good in cutting and pasting. Cut and paste the receptor binding domain of the pangolin coronavirus and became the magnanimous SARS-CoV-2 virus. jumped into the humans and started killing people. That is the natural origin virus.
Yes.
But scientists started questioning. Now, if rat G13 and pangolin coronavirus to combine become SARS-CoV-2, they should be already existing in nature. The problem is the first publication of the rat G virus and the pangolin coronavirus happened after the onset of the pandemic. So scientists started questioning, oh, you have got two pre-existing viruses combining to form a new virus, but we never knew about the existence of rat G13 or this pangolin coronavirus, this particular pangolin coronavirus, because there are different types of pangolin coronavirus. And then this various scientists started questioning, oh, this is not right, that is not right. Now, what are the evidence or possible suggestions of lab origin? I will not say 100% evidence, but these are the suggestions what some scientists believe it could be of lab origin. Now, several scientists believe this is not a combination of rat G13 and pangolin coronavirus. It's a combination of another bad coronavirus called as ZC45. ZC45. And the receptor binding domain of SARS-CoV-1. What is SARS-CoV-1? SARS-CoV-1 is the one which killed 1,000 people, affected many countries in 2004, originated in China, the SARS, the original SARS.
Yes,
and some scientists believe it's a receptor binding domain of SARS-CoV-1 cut and pasted into the backbone of ZC45.
Okay, and what are the basis for that? What is the basis for that assumption, doctor? Without getting too scientific, just in layperson terms.
Because first, the natural origin theory people believe the rat G13 backbone has got 96% genetic level similarity with SARS-CoV-2, and the receptor binding domain has got 100% similarity with this pangolin coronavirus. But some scientists believe that particular pangolin coronavirus, yeah, it's not a real virus. They believe it could be a fabricated laboratory sequence, because you can fabricate a virus. You can cook up a virus on your computer and even create a virus. Because whenever virologists discover a new virus, they upload the genetic sequence onto a global database called GenBank. Anybody, all scientists do that. They give, and that's why you get a name for the virus. And to get a name of the virus, you don't have the full 30,000 sequence. There is a particular segment called as RDRP. It's a very small segment. You need to upload, it's around 500 letters. You need to upload only that to get a name. You don't need the full sequence. So scientists believe that, the receptor binding domain belongs to SARS-CoV-1, but not a perfect cut and paste. They probably modified the certain changes in the receptor binding domain, so it doesn't look like SARS-CoV-1. Now, how do you make that? If you make changes, don't you think it will affect the function? If it's not functional, how can it infect humans? So some scientists believe that changes are so ingenious, so intelligent way that if you look at the genetic letters, they look different, but look at the protein function, exactly the same. So there are ways of doing it. I'm not explaining that. That's why I explain. You can change the protein by changing the genetic codons, because I mentioned the 64 triplet codons and 20 amino acid. So if you want to change, you can have the same amino acid by changing the letters. Still, the letters look different, the protein may look the same. And there are other ways of making changes in the protein without affecting the function, but changes the appearance. Scientists believe with acceptably good scientific basis that it's a cut and pasted SARS-CoV-1 receptor binding domain put into the backbone of ZC45. It needs a lot of discussion. Now, I will come to one important point. Why can't we believe, even if this is different virus, it would have happened in the nature. But scientists believe that there is evidence of cut and paste by human beings.
Oh, okay.
And how do scientists, just like cut and paste happen in nature, you do it in the laboratory, and virologists do it every day.
So
they do it every day. And the knife and the knife and glue used for cut and paste, It's a very common knife and glue used in virology laboratories every day by every scientist.
Oh.
Yes. Does it happen in nature? Yeah, nature also the same glue and paste is there. But it's extremely unusual for the same common daily glue and paste coming exactly on both sides of receptor binding domain. But still you can claim, oh, Mother Nature is so magnanimous, so able, even rare cut and paste can happen on either side. Mother Nature can do anything. But then the argument is, see, the Dr. Shi Sengli is the chief of the virology in Wuhan Institute, and she is the leading coronavirologist in the world. And Shi Sengli and team in 2008 have published paper on cutting and pasting of other viruses using the same technology. It's available for everybody to use. So this is something unusual to happen naturally, can happen, but unusual, but daily happening in virology laboratories, and the same scientists in the Wuhan Institute of Virology and other scientists in the world have used the same cut and paste, use the same knife and scissor, and publish papers on recombination.
Right, just for the audience, if I can just break this out down, simplify it. Basically, what the doctor is saying, and please correct me, doctor, if I'm getting it wrong, is that there are certain techniques where you can cut and paste a virus to create a new variant or a new virus. And this technique is something that one of the chief doctors, a leading virologist from the Wuhan lab, has actually written about this technique a couple of years ago. And it's something that's followed across labs. So the fact, what it does is really it lends credence to the theory or the thought process right now that this could be lab created, because it ties in much too closely to the expertise that this Wuhan lab possesses. Am I correct, doctor?
Yes, you are correct. And I will explain one more point. If you analyze the genomic sequence, there are dozens of potential evidence of lab origin. But I'll give you one more point. Between the rabbit ears, there's a cleavage.
Right.
And in the cleavage, there is something called as polybasic furin cleavage site. You don't have to remember that. There's something called a furin cleavage site in the cleavage. But there are other viruses with exactly the same polybasic cleavage site. The Middle East coronavirus has one. And there are many other viruses with the same cleavage site. But if you analyze the polybasic furin cleavage site of SARS-CoV-2, it has got a rare sequence that never exists in any natural coronavirus. I explained to you, there are multiple triplet codons can code for one amino acid. There is one amino acid called as arginine. So if you want two arginine in your protein, you need two triplet codon sequence. And arginine, the protein, the amino acid, can be coded by 6 types of triplet codon. So if you need arginine, you have got 36 possibilities to create 2 arginine. 36 possibilities. But if you look at this virus, in the polybasic furin cleavage site, there are 2 arginine amino acid coded by exactly the same 3 letters.
Public domain now, but there has been a lot of back-end work in the virology community.
Oh, what I explained are just 2 simple examples. You can analyze the genome, there are dozens of examples of potential laboratory origin. Plenty of data is published, but rather than supporting such evidence, the famous virologists came forward and criticized that all this evidence are childish. But that is a story of the past. Situation is changing. At least now the virologists are coming forward and thinking the possibility of a laboratory origin. Now, as I explained before, even if it is a laboratory origin virus, yeah, it's not just the China. The virologists, military laboratories, scientists do it in several countries. And if you look at the SARS-CoV-2, even if laboratory origin, it's just a baby chimera virus. We call chimera. The cut and paste recom in the lab is called chimera. This is just a baby chimera virus. The virologists in the military laboratories in several countries have stockpiles of virus far deadlier, far dangerous than SARS-CoV-2. What with SARS-CoV-2 is a baby virus. So even if it is lab origin, I'm not surprised at all. There are several countries, I'm sure the Wuhan Institute of Virology, even this is lab origin, they will have a more dangerous virus. And military laboratories of several countries have far more dangerous virus. How do we know that? There is published data available. scientists have earlier published the chimeric viruses cutting and pasting from SARS-1, and scientists have published such virus and they have done experiment on human cell lines to know whether it is working or not. So this is all well-known this recombinant gene technology of recombinant virus technology, which is well known for the scientific community. There's nothing unusual about it. And virologists are very, very intelligent group of scientists. They know how to cut and paste and they know how to hide their cutting and paste. But in the case of SARS-CoV-2, there are potential evidences of laboratory origin. But as I explained earlier, there's nothing unusual about it.
Yeah, doctor, so basically it is like every crime, there are fingerprints. So or now in today's time, DNA, so you can analyze that if it's CSI. So with this as well, there are some fingerprints, there is some DNA that can be forensically analyzed and conclusions can be reached, is what you're essentially saying. And now from the perspective of the general public, what lies ahead, assuming just by chance this is a lab created virus? Does it make it easier for scientists to find a cure, a vaccine that is 100%? Is there any benefit in terms of finding a way out? Because right now it just seems like a long dark tunnel where we're all locked in. Our lives have changed dramatically. And staying locked in is actually a luxury because so many people, like you mentioned in your opening comments, don't have that luxury of staying home and have to go out and work and expose themselves to risks. So what is the end game here?
Let us look at the implications for the public and the scientific community. As far as the public is concerned, it immediately doesn't make any difference whether the virus is of natural origin or laboratory origin, because the people are getting killed. People are suffering because of the virus, whether it is of a natural origin or laboratory origin. Whatever may be the case, we need drugs, we need vaccines. The best way of coming out of the pandemic is, one of the best ways is vaccinating fast, because vaccine is our most efficient solution in preventing the spread of virus. So personally, as a man of science, I recommend the scientific community and recommend the public to get vaccinated fast so that somehow we have to come out of the pandemic. It may be of natural origin, it may be of lab origin, but getting vaccinated fast, following masking, social distancing, hand washing, and all the other precautions we need to follow, including vaccination. But that is for the public. Now, for the scientific community, we need to consider, at least discuss the possibility of a laboratory origin. We don't have to reach any conclusion. It will be very difficult to reach a conclusion. In my opinion, whatever investigation you do, whatever investigation done by WHO or Wuhan or UN or any countries, will never reach a conclusion that it is of laboratory of natural origin. It will be always a doubtful two theories. But that's okay. What we need is, we don't need conclusion. We need a possibility of a laboratory origin and ways to solve that possibility of laboratory origin in the future. How can we do that? Scientific community should be open about the discussion. If you don't do that, the future generations will criticize the scientific community, today's scientific community, medical community, biotechnology community, the medical journals, and the whole scientific community may be questioned by our future generation. Suppose the world scientific community did not come forward in the generation of nuclear non-proliferation treaty. How will we, the current generation, would have looked at our previous generation who created the nuclear weapon? Currently, we don't blame them. We don't blame those scientists because we know what happened. Later, we know the scientific community was shocked by the impact nuclear explosion made. Albert Einstein, who wrote to the American president for the creation of the nuclear weapon to beat the Germans in the war. Later, after seeing the impact, what created in Japan, Albert Einstein told, he was shocked. He was shocked. and he basically regretted his advice on generation of a nuclear weapon. And then the world of science changed their opinion. They came forward. That's what we need. We don't need proof. We don't need to finger point at few scientists. We don't need to finger point at few laboratories. We don't have to finger point at 1, 2, or 3, or 4 countries. We need to discuss the possibility, a remote possibility at least, of laboratory origin and find ways so that the gain of function is stopped forever. We don't want Frankensteins. We don't want baby Frankensteins. We don't want elephant Frankensteins. We don't want chimeric Frankensteins in the world. We need a treaty signed by all the countries, the military laboratories across the world must stop gain of function research, even for the ethical purpose, for generation of vaccine and drug. We don't need chimeric viruses. We don't need gain of function research. And number 2, all the existing stockpiles of chimeric viruses must be destroyed, because several countries have stockpiles of chimeric viruses. All these stockpiles must be destroyed. Just like we reduce the number of nuclear weapons in many countries as a part of treaty, we must not reduce, destroy the stockpiles of chimeric virus, and no future result. Yes, we may have an explanation. We probably could have generated this virus for the betterment of humanity. But did it help the humanity? Made the humanity?
No.
If it is of lab origin,
I mean,
what we created with a good interest destroyed the humanity.
It feels like a Marvel comic, where that wicked mad scientist sort of stereotype emerges in terms of just how frightening it is. And I'm speaking as a layperson, doctor. I don't want to position or project myself as anyone that is particularly informed on this subject, but just as seeing the kind of devastation this virus has caused across the world in our own personal circles. I can't think of anybody, I'm sure you have as well, I have lost close people to this dreaded disease. And we just want it to end, and I 100% endorse what you're saying. If there is to be a global movement after this, it has to be one that takes control of this kind of experimentation, which can cause such devastation around the world. Doctor, it's really, I'm grateful to you for coming in, for making something that is so complex. It's still very complex, even when you describe it, but it becomes a little more understandable for us, because most of us, we feel very helpless right now and feel trapped in this situation where there is seemingly no end in sight. And every time something new comes up, there's a variant, which is even more transmissible. It feels like this is the new normal and it's going to continue for a while. So I want to first, of course, thank the good scientists, who are working hard around the clock to figure out what this is and how to get humanity out of its clutches. I can't even begin to imagine the amount of rigor and sacrifice and intellect and hours it takes to be able to decipher this, to understand it, to battle it. So my gratitude, my immense gratitude to the good scientists of the world who are doing this. And my hope, my earnest hope, and thank you for sharing that with me and really opening my eyes to it, is that the global community comes together and takes in hand this kind of flagrant misuse of science. And that's really the way I see it at this point. But Doctor, just if you could once more just tell us about this gain experimentation that you've mentioned a couple of times.
The gain of function research means you take a virus and do genetic manipulation in that virus so that that virus behavior changes. Gain of function. For example, that virus become more transmissible. The ability of that virus to infect more number of people changes. Or the ability of that virus to kill people changes. So it's a gain of function. That function may be either infectiousness or lethality. And why do we do that? Because I actually explained, Mother Nature is doing it on a daily basis. In fact, one of the biggest proponents of gain of function research once told, The biggest terrorist in the world is Mother Nature, because she's keeping on creating dangerous chimeric viruses. So what can we poor human beings do? Get ahead of the Mother Nature, create more dangerous chimeric viruses, at the same time creating solution to it. Now, why do scientists want to get ahead of the nature, Mother Nature, who is a terrorist? Because you become a bigger terrorist, at the same time find a solution to it. By the time Mother Nature releases her virus, we'll be ready with the weapons to fight it, like drugs, vaccines, equipment. So the intention is good, but never ever mess around with the Mother Nature. All the story of Frankenstein really probably is coming true today. Never mess around with the Mother Nature. We are not gods or goddesses. We are humble creations of the Mother Earth, the Mother Nature. Respect the Mother Nature. Don't mess around with her. If you mess around with her, probably such incidents will happen. It's a lesson for us. Let's not repeat it again.
Couldn't have ended on a better note, Doctor. Don't mess with Mother Nature, because she can strike back in ways that we won't be able to handle. And thank you so much. It's been brilliant talking to you. And I'm sure that this video is going to go far and wide, because there hasn't been quite such a discussion and such an elaborate explanation of the circumstances surrounding the origin of the virus. So I thank you again, Dr. Abdul Ghafur, for joining me. Yes, Doctor?
One more point I missed out. It's important for the audience.
Oh, sure.
About the origin of virus. I'll say that, you can edit maybe in between. Because in epidemiology, we call a phylogenetic tree, where we map the different variants of the virus. Now, you all know alpha variant, beta variant, gamma variant, delta variants. We map it on a tree, and that's called as phylogenetic tree. Now, if you analyze the phylogenetic tree of SARS coronavirus, first one, SARS-CoV-1, 2004, originated in China, or the Middle East coronavirus happened in Saudi Arabia 2014, or any other major virus outbreak, there is a specific pattern. The virus outbreaks, if you chart the plot, it starts as roots. Then it becomes a single line like a trunk. And after that, the branches start appearing. What is the basis behind it? Because, for example, if this mixed up bad corona, pangolin coronavirus and pangolin, creation of SARS-CoV-2, it jumps to humans. But it jumps to humans, it cannot immediately create a pandemic. It has to get adapted to the humans by passing over in multiple humans, so that appears as roots, different clones of the virus. All the clones cannot produce epidemic or pandemic. Slowly they get adapted to the humans. Only that particular clone, which is well suited for the humans, become an epidemic and pandemic. So it will appear like roots, multiple line, become, one of them become prominent like a straight line, like a roots, and then the big trunk of a tree. And then after several weeks or months, the variants like alpha, beta, gamma, delta comes appearing as branches. So a natural epidemic appear like roots, become a trunk, then become the variant branches. If you analyze the SARS-CoV-1, root, trunk, branches. Middle East coronavirus, root, trunk, branches. Analyze the tree of SARS-CoV-2. There is no root. There's just a trunk in the beginning, and then after some time, the branches. No root, big trunk, branches. Then where are the roots? The roots can be, the roots has to be either in humans or animals. There is no evidence of roots in humans or animals. So people claim that the roots are in the laboratory. What we saw is trunk in the humans and then the branches in humans. That could be probably true. Again, we are coming back to the basic point. Even if it's lab origin, we don't need this to happen in the future.
Yes, absolutely. Thank you, Doctor, once more.
It may be an important point for the audience.
I'm glad you brought it in at the last minute because I think it's so visual that I think it puts everything in a nutshell. So thank you again, Doctor. Namaskar. Thank you for joining.
You can cut and paste this. Namaskar. Namaskar. Okay. Maybe you can paste that. I explained that.
Yeah, cut and paste is your favorite word for the day.
Yeah. So what you can do is, I explained 2 laboratory features. The receptor binding domain cut and paste and the arginine GCC cut and paste. After that, probably you can cut and paste this tree idea.
Or we'll just let it flow. Thank you so much, Doctor, and thank you to all our viewers for watching. And I hope to have you back on the show again to discuss vaccines at some point because there's a lot of conversation on that also that's happening. And I want to do something on how we, as the consumer in this case, can tell the different vaccines. Because when you say mRNA, till 6 months ago, I would have not known what you're talking about. But because of Pfizer being an mRNA vaccine, I know what you're talking about now. And I know that Covishield is a vector. So, just to kind of put that across to laypersons, it would be most helpful. I hope you can come back for that.
Sure.
Okay. Thank you so much, Doctor.
Great.
Bye.
Great. GCC. Yeah, you can, because this polybasic furin cleavage site is a very important for the disease pathogenesis.